Hi everyone, welcome back to my channel, and today we are going to learn about translation. Translation is a process of synthesis of this protein from mRNA. And before going into the topic, we should know what is codons, what is the structure of tRNA, and what are these ribosomes. So first of all, let's learn about these codons. Codons are the sequence of three adjacent nucleotides in a strand of nucleic acid that's code for a specific amino acid in synthesis of protein molecules. For example, this is a mRNA, and there is three adjacent nucleotide AUG. So this will code for a specific amino acid, that is methionine. And the features of codons are specificity, that is genetic code is specific. A specific codon always code for the same amino acid. Another is redundancy, that means an amino acid can be coded by more than one codon. That means, for example, there is AUU, AUC, and AUA. These all codons will code for amino acid isoleucine. That means this AUU always code for isoleucine. This AUC will also always code for isoleucine, and AUA will also always code for isoleucine. But these isoleucine have three different kind of codons that will code for it. Another is universality, that is a codon codes for same amino acid in different organism. For example, in prokaryotes or eukaryotes also, this codon will specify the same amino acid. Another is non-overlapping. It means, for example, there are codons A, B, C, D, E, F, G, H, I. So these are non-overlapping. This ABC will code for one amino acid, then DEF will code for another amino acid. Likewise, GHI will code for another. There won't be any BCD or CDE. This is the codons will be non-overlapping to each other. There are total 64 codons. One is the start codon, that is AUG, and another three are the stop codons. The three stop codons are UAG, UGA, and UAA. Now let's learn about the structure of tRNA. tRNA is the transfer RNA. It is synthesized from RNA polymerase 3. The primary structure, that is the nucleotide sequence of tRNA molecules, allows extensive folding, and these are complementary to generate a secondary structure that appears like a clover leaf. So this structure looks like clover leaf shape, and this is the secondary structure of tRNA. tRNA have different measure. They have four arms. One is the acceptor arm, another is anticodon arm, third one is this D arm, and the fourth is T arm. There is one extra arm also, which is also known as variable arm. So first of all, let's talk about this acceptor arm. This is capped with a sequence CCA, where amino acid is attached. And another is this anticodon. It contains three specific nucleotide, that is the anticodon. And this is responsible for recognition of triplet codon in mRNA. Another is dRNA. Why it is called dRNA? Because there is presence of dihydrouridine, and it is important for recognition of addition of amino acid. And another is the tRNA. This T signifies thymine, this Shi signifies pseudouridine, and C signifies cytosine. It involved in binding tRNA to ribosomes. So this is the basic structure of tRNA. Now let's talk about ribosomes. Ribosomes are found free in cytoplasm also, but some ribosomes are bound to endoplasmic reticulum and forms the rough endoplasmic reticulum. This contains rRNA and proteins. And these ribosomes are always arranged as a large subunit and as a small subunit, and they are different in case of prokaryotic and eukaryotic cells. In case of prokaryotes, there is 70S ribosome, and the small one is the 30S, and the large subunit have 50S ribosome. Likewise, in case of eukaryotes, there is 80S ribosome. The small one is 40S and the large one is 60S. In this ribosome, these ribosomes have 4 sites. One site is for mRNA, and this is a small subunit. In this large subunit, it have 3 different sites. One is this E site, another is P site, and the last one is A site. This E site name is exit site, P site name is peptidyl site, and A is for amino acyl site. This amino acyl site is for the entry of new tRNA. P site, the function of P site is it helps in growing polypeptide chain. And E site is through which our tRNA will leave the ribosome. Now let's learn about translation. It have mainly 3 steps. First one is initiation, second one is elongation, and third one is termination. In translation occurs in cytoplasm, and the main aim of translation is to join the amino acid and formation of polypeptide chain. So in first step, that is the initiation step, there is mainly 2 main things occurring. One is activation of amino acid, and second one is transfer of amino acid to tRNA. So we should know about one enzyme, that is amino acyl tRNA synthetase. What does it does is this amino acid binds to tRNA with the help of this enzyme amino acyl tRNA synthetase in presence of ATP, and it will form amino acyl tRNA plus AMP plus 2-phosphate molecules. This amino acyl tRNA synthetase function is recognition of suitable amino acid and tRNA and transfer of amino acid to the acceptor arm of tRNA. We have already discussed about the structure of tRNA. There is the acceptor arm, so this enzyme will join this amino acid to the acceptor arm of tRNA. And we have already discussed about this anticodon arm also. The anticodon present in the tRNA will join to the complementary base sequence of mRNA. So this is our mRNA and have codons. For example, there is codon AUG. This tRNA will, the anticodon present in tRNA will come and base pair with mRNA, which have its complementary base sequence in P site. So in this P site, there comes the initiator tRNA, and this AUG code for the amino acid, that is methionine. So what happened in the process of elongation, this ribosome have 2 subunits. This is the larger subunit and this is the smaller subunit. They are moving forward. So there comes another tRNA which have anticodon CAG, and it codes for complementary base sequence of mRNA which have GUC. It also codes for another amino acid. So this amino acid and the amino acid present in the P site will form a polypeptide bond. So this is polypeptide bond. And this polypeptide bond is formed by rRNA. After joining, this methionine will go in this A site. So there in this P site, this tRNA won't be having any amino acid, there will be empty. Now this RNA, this ribosome is moving forward. So while moving forward, this AUG will come here in E site. So here this AUG comes in E site and it leaves, as it is the exit part of our larger ribosome, this tRNA will leave the ribosome. Now again there will be another tRNA that will come and that will code, for example, of ACG. So it will base pair with complementary base sequence of mRNA, that is UGC, and likewise they will form the polypeptide bond, and again this amino acid will move in this A site. So this process will continues until there is something known as stop codons. So the stop codons are UAA, UAG and UGA. So whenever stop codons come in this messenger RNA, there will no tRNA will come here. So there comes one important thing, that is the releasing factor. This releasing factor will dissociate the larger and the smaller subunit of ribosome. And also break the polypeptide chain. And it will release our proteins. So there will be formation of our polypeptide chains in cytoplasm. This is how the protein is synthesized. So this is all for today. Hope you guys enjoyed. We will meet up in our next video.